Hello, everyone, and thanks for joining me today for this webinar on FlowSOM in FCS Express 7. I'm super happy to take care of this webinar and to be with you today. Online with me, Kaya Ghosh, will take care of answering any question you will type in the question section. So take your time to navigate in the question section of the GoToMeeting webinar, and we will reply to your question in real time. Okay, so let's see the agenda for today. As usual, I want to provide you with an introduction on the topic. So we will see why we need clustering, what's a clustering, and what are self-organizing maps, which is the core part of the FlowSOM algorithm. And then we will see how to run FlowSOM in the latest release of FCS Express 7. And this also allows me to present you one of the new features in FCS Express 7.6, which is the pipeline. So pipeline will give you access to FlowSOM, but also to a lot of many other high-dimensional data analysis tools like t-SNE, UMAP, optimized t-SNE, SPADE, K-means, hierarchical clustering, and you will see it's really amazing. So why do we need clustering, and generally speaking, why do we need high-dimensional data analysis tools? Because nowadays, flow cytometry, but actually I would include, of course, mass cytometry, definitely image cytometry, generate data with really a lot of parameters, a lot of features, so a lot of dimensions. And the standard way to analyze this type of data, and with standard way, I mean the opening multiple plots with two-dimension plots, it's not really efficient. It's time consuming anyway. What can you retrieve from like this example with like 200 different plots? It's really non-human readable. If you imagine to look into your FCS file, you will see something like a table in which you have events in rows and fluorescences, so parameters, also scatters, let's say, in columns. So rows are basically observations. Columns are usually called like features or dimensions, okay? Because you see, if you have like FL1 and FL2, you can plot your cells in a 2D plots. If you have also FL3, you can plot in a 3-dimension plots. With more dimension, you cannot use any plots. And this is the reason we need dimensionality reduction and clustering. Dimensionality reduction acts by reducing the number of dimensions. Okay, this is what t-SNE, UMAP, PCA do. Clustering, on the other hand, reduce the number of observations. So they reduce the number of rows, not the number of dimension, but by default, the number of rows. And they do this by grouping similar events together. So you may have like 1 million of events, and maybe the clustering algorithm finds 10 cluster. Okay, so instead of having a table like this with 1 million of rows, you will just have 110, 1000 rows if you have 110 or 1000 clusters. Example, of course, are FlowSOM. This is why we are here today, K-means, SPADE, Phenograph, and many other. Now, this is the definition of clustering. So clustering is grouping data in a way that data within the group, so within the cluster, is more similar to each other than data that are in other groups. So if you look at this picture at the right, you have, I mean, it's really clear that you have like 3 population, right? So if you ask like K-means to find 3 cluster, those 3 cluster will probably be like what you see here in the picture, like one cluster that group all the green, one cluster that group all the red, one cluster that group all the black. So the idea of the clustering is that then once you have defined the cluster, so once you have assigned every single event to a cluster, okay, we will call those nodes in FlowSOM, but they are clusters. You summarize all the events in that cluster with a unique point, a unique summary. It's usually like the center of those cluster, okay? You can think about the MFI of that cluster. So you see, you're not reducing the number of dimensions. So each of those clusters still exists. For example, the green one still exists in dimension 1 and dimension 2, okay? Simply, we reduced the number of events that we were dealing with. Now it's 3 instead of maybe 3,000. Another key point is that clustering doesn't really tells you anything about the biology. I mean, the clustering is grouping similar events together, then it's you that you need to label a population, a cluster, as, I don't know, CD8, Treg, or B cells. There are tons of type of clustering. Okay, you see here at the very bottom of the slide, FlowSOM, it's part of the partitioning clustering, but then there are really a lot of other type. Now, SOM, so the self-organizing map, not the FlowSOM. SOM, it's a technique that have been developed like 30 years ago, actually September 1990, by Professor Cohen, and it's considered a clustering and a dimensionality reduction. So you will see why this, okay? It's not only a clustering, it's also dimensionality reduction and a visualization tool. It was really popular, and it's part of the competitive learning. It was really popular for study neural network, because as you will see, it's really working like neurons. So neurons in the real world compete to access the stimulus. And as you will see, the nodes of the self-organizing map compete each other to access the target, the input. And the input, of course, is your data set. Now, before starting, just have a look at this animation, okay? You have some, let me draw here. You have some population, you see here. Those are all the events, let's say, of your data set. And in your case, those are biological population, okay? And those exist in a multiple dimension. Now we have just a 2D representation, but they exist in like 20 parameters when you have a 20 parameters, 20 color experiment. And you start at the beginning by creating a grid. Okay, this is the self-organizing map. Okay, you decide which is the side of the grid in the size. In this case, it's a 5 by 5, and you decide where to place this grid. Okay, in this case, this grid is placed more or less in the center of your data set. Now, each of the nodes of this grid are basically centroids. Okay, so if you want to think about k-means, maybe you are more, let's say, habits. You always or dealt in the past with k-means. Those are like centroid, but in contrast with k-means, in this case, these nodes are connected. Okay, you can see that every node is connected with some neighbor nodes. Okay, in this representation, what you see here, it's a rectangular grid. Nodes have at most 4 connection. Okay, this has just 2, this has just 3, this has 4 connection. Okay, so the idea is that you will stretch, okay, you will shape this grid, okay, in order to mimic the overall data set. So let me now play this small animation. You will see that this grid is stretched step by step. Okay, every single movement is one step. Okay, at the end, what you want to have is a good representation of your data set. Now, the beauty is that this grid exists in 2 dimensions. Okay, so with this grid, you have a 2-dimensional representation of your multi-dimensional data set. This is why SOM, it's not only a cluster algorithm, but it's also a dimensionality reduction. Oh, sorry, just pressed the wrong one. Now, how does SOM do this? Okay, there are several different, let's say, calculation. The first one is the initialization. Okay, let's take this this example. This is like FL1 and FL2. Let's say I have here 6 points, 6 events. Okay, it's a quite small data set. I define my grid. In this case, it's not 10 by 10, it's like 2 by 3. So it's a 6-node grid. And then once I've defined the size, okay, I need to define where to place this grid. Okay, before we place the grid within the data set, okay, that's probably the best. Okay, in this example, in this slide, the grid is placed here. Okay, it's at the left of my points, but that's fine. It's just It's up to you, basically. After the initialization, you pick up one random event from your data set. Okay, it means one random event from your cells. In this case, it's this one. It has been blue labeled. And then you calculate the distance between this and every node. Why this? Because you want to find the winner node, which is the closest. Okay, and as you can see in this example, the closest one is this one, is the bottom right. Once you've done that, you start stretching your grid. Okay, so step 3 and step 4. Step 3, it's called updating of the winner node. The winner node is the red one. It's the closest to the randomly selected events. So the idea is that you start moving this towards the selected events. And the idea is that you are not covering the full distance. Okay, these red nodes will not overlap at the end of the movement with the blue nodes, with the blue events. It will just cover a fraction of this distance. It can be like 10%, it can be 20%, 50%, 90%. It can be whatever. So this fraction, it's called learning rate. It's also called alpha in this algorithm. The winner node doesn't move alone. Okay, once the winner node moves, okay, let's move it. So this is step 3. Okay, in this case, it covered probably like 50 or 60% of the distance. The winner node pull with itself a bunch of neighbor nodes. So the winner node is not the only one that moves. And the idea is that the closer a node is to the winner, the more it will move. And on the other hand, the farther a node is, the less it will move. So now let's let those nodes moving towards the blue points, okay? Those events will move more than those, and those events will move more than this. Basically, you will see these last events will not even move at all. It's too far. So let's see the animation for step 4. Here we go. So you see, let's do it again. So this was after step 2. Then we update the winner and we update all the other nodes. So this is just one cycle, okay? This is just the stretching the grid towards one single event, the blue one, in our data set. We need to do this a lot of time, like 10,000, okay? So basically, it's an iterative process. You can see in this animation, the grid, it start to be like a rectangle, and then you start stretching from the corner, okay? Every time, at every step, you select one node and you move the grid. And maybe at the next step, you select this node and you move the grid. And you see that those nodes, okay, move more, okay? They cover longer distance than the nodes here, because the nodes here are far neighbor, let's say. And you can also see, now I will start the animation, that basically this node, it's too far. It will not move at all in this animation. Okay, you see here. So again, in this case, you have like 3 population in a 2 dimensions, but in your case, you will have, I don't know, 10 different population, 50 different population, biological population, or 50 in a multi-dimensional space. But the grid is doing the same stuff. Now, how do you define how much to move every single node? Okay, there is a formula. Okay, unfortunately, if you want to, let's say, go through the algorithm and know how to deal with a different setting, we need to go through this formula. But you will see there is nothing to be scared. So the movement, okay, so the space that the nodes, each node cover towards the blue point, it's called learning. Why learning? Because it's a way to, let's say that your grid, it's mimicking your data set. This is based on the actual distance between a node and the target, okay, in this case, between the red and the blue. And then it's defined by 2 different variables. One is called learning rate, okay, we already see that, okay, remember, in our example, it was basically like 60%, something like that. Okay, so let's say 0.60. And the other one, it's called neighborhood function. It's called also h. So those two values, alpha and h, can be are included between 0 and 1. Okay, usually they are lower than 1, and this means that usually the learning it's smaller than the actual distance, because the distance is multiplied by something which is lower than 1. Not only, but those two guys, the alpha and the h, so the learning rate and the neighborhood function, are decreasing function. So at every cycle, okay, remember what we saw in the last slide, this is just point 1 and then you stretch towards that point. But at cycle 2, you will pick up another point and you will stretch the grid towards that other point. At cycle 3, you will repeat that for another point. At every cycle, alpha and h decrease. The idea is that the movement of your grid, okay, the learning, decrease at every cycle. So your grid is basically getting less flexible, okay, and it's more stable, in other words. Also, you don't want to move the grid ab infinitum. Okay, at some point the movement will be 0. So this will be 0, this will be 0, and at the end the learning will be 0 and everything will stop updating. That's the end of the calculation. Now, those two guys can decrease in two different ways. They can decrease gradually, like you see here on the left, linear decay, or they can decrease with this asymptotic decay. It means that they decrease a lot immediately and then they are basically flat. Okay, so in the first case the flexibility gradually decrease. In the second case, asymptotic decay, the flexibility drop down after few iteration, and then it's basically stay quite stable. Okay, so the alpha is basically just one value. Okay, it's called learning rate, is defined by the user define which is the initial and the final value. Okay, if it's 1, okay, the software will calculate 1 like as a starting point, 0 as a final point, and it will automatically decrease it from 1 to 0. But the user can select whatever, can use like 0.1 as a starting point and 0.05 as an endpoint. Okay, of course, FlowSOM has some default value, so it's up to you whether to change them or not. The h, so the neighborhood function, it's more, let's say, I would say complex. Is not just a number, it's an actual function. Actually, also the alpha, it's a function, as you can see below. But anyway, the h, it's called neighborhood function and dictate how the other nodes, not the winner nodes, moves. So the neighborhood function, it's always equal to 1, okay, for the winner. This means that at the end, the winner only rely on the alpha. Okay, but for all the other nodes, the h, so the neighborhood function, has some value, which is usually lower than 1. So the movement that they are going through, it's lower. So the learning is lower. Now, let's see which is the, let's say, the main point of the neighborhood function. So you need to define a way in which you say, okay, all the nodes in my neighborhood, okay, the nodes that are close to my winner, okay, will move in a certain way. All the other one will move less, or either they will not move at all. Now, you need to define a sort of boundary, okay? You need to define the definition of neighbors, or of neighborhood, sorry. And this is the neighborhood spread, okay? This is the value defined by the user, and it's really the boundary of the neighborhood. You can say the neighborhood in my for my friends is, I don't know, 1 block, and everyone which is out of the block, it's still a friends, but it's not formally a neighbor. And everyone which is within my block, it's actually a neighbor. So this is a clear, let's say, cutoff. Okay, this is called boxer function, or also bubble function. If this is, have a look at this example. If this is the winner node, okay, you really draw a circle around and you say everything within this will move exactly as the winner node, okay? Basically for all the nodes in the blue area, H will still be 1. All the nodes that are outside the area will not move at all, okay? It will be 0, okay? This is either on or off. And if you look at the graph at the bottom, okay, what you see on the x-axis is the distance between the winner. So nodes that are within this region, okay, they will move. Everything which is outside will not move at all. Again, this is on or off. Or you can be more, like, say, flexible and gradual and say, again, here on the X, you have the distance between the winner node and every other node in the 2D grid. And you can say, okay, the dots that are, the nodes that are at a certain distance, let's say 1, will move with this neighborhood value. Nodes that are farther will move less, and at some point you will reach a value which is pretty close to 0. Okay, those are two different ways of calculating the neighborhood, whether you want to be really strict or whether you want to be more gradual and flexible. So again, just to summarize, this is how you calculate the movement for each node. This is the distance between every node and the selected point. This is the learning rate. It's defined by the user. And this is the neighborhood function. And the neighborhood function can be either this way, on and off, or can be Gaussian. Okay. Now, FlowSOM. FlowSOM rely on self-organizing map. It has been published in 2015 by Sophie van Gassen, and it's basically the application of SOM in flow cytometry, or actually in cytometry. You can also use it for mass cytometry, image cytometry. And what FlowSOM is doing is also representing in a very nice way all the nodes. Those are all the 100 clusters generated by FlowSOM in a 2D tree-like representation, which is pretty similar to SPADE. Actually, the calculation is the same. It's a minimum spanning tree. Now, FlowSOM formally perform 3 main, let's say, type of calculation. The first one is running the clustering with self-organizing map, which is this, is exactly what we saw before, okay? Then it built a minimum spanning tree, and we will see what does it mean. But basically the result of the minimum spanning tree is this sort of representation in which each node of the FlowSOM grid, of the SOM grid, it's connected to the closest neighbor. Okay, we're no longer speaking about events. Okay, still is the closest neighbor in the multidimensional space, but we are speaking about clusters. So each of those nodes, it's one of the clusters that you see on the left in the 2D grid of FlowSOM. And the last step, which is really cool, it's a consensus clustering. This was one of the main limitations in SPADE. Okay, so if you look at the representation that you see here in the center, you can imagine that, okay, you can color, for example, each node based on the expression of a given antigen. Okay, so you can say that those are clearly green positive, those are light blue positive, those are red positive, and so on. But sometimes it's a shade. Okay, sometimes it's really hard to say those are an homogeneous population and those are not. So it's hard to put some boundary. And with consensus clustering, you can cluster clusters instead of events. So the idea is that you cluster those nodes and you group similar nodes, not similar events, together. And this is depicted in the FlowSOM tree with this halo. You see there is this red, orange, green. I mean, the color it's just fancy, but anyway, same color means same meta cluster. Okay, this is really useful because the idea is that with the meta cluster, you increase the chance to, let's say, really identify some biologically relevant population. Now, let's do this. Let's see how we can do this in FCS Express. So in FCS Express, FlowSOM, it's one of the many algorithms that you can run through a very new feature in FCS Express. We just released it last week. This new feature is called Pipeline and gives you access to a wide variety of algorithms. So a pipeline, generally speaking, is a set of processing elements, usually mathematical calculation, that are connected in series, as you can see here at the right, like A, B, C, and D. The key point is that the output of one step is usually the input of the next one. So A generates some data, and this data is the input for B. B generates other data, and this data is the input for C, and so on. Pipeline are very well known in the like in the mathematical, statistical, bioinformatician field. They are basically at the basis of every programming and computing script. Now, I would say all of the algorithms are pipeline themselves, like FlowAI, Disney, Optimize Disney, UMAP, K-means again. And the beauty is that with pipeline, okay, you can combine all those algorithms. So you can combine pipelines together. So a series of steps, which is formally a pipeline, okay, can be considered as a step in a wider pipeline. This is something that you cannot do with other software. So a lot of other software allows you to run like FlowSOM, Disney, FlowAI, Disney, FlowSOM, UMAP, but they allows you to run them as a standalone algorithm. You cannot really combine them. So with the pipeline, you can really combine them. Today, we will focus on FlowSOM, but if you are interested, just navigate on our website and there is a recorded webinar about how to use pipeline and create a pipeline with multiple algorithms. So pipeline apply perfectly to the workflow in high dimensional high-dimensional data analysis in cytometry, where you start with some FCS file, then you do some data cleaning, then you do some pre-processing, maybe you want to scale, to normalize, to downsample, and at that point, you start running your algorithm. Okay, FlowSOM is just one of the many. So pipeline in FCS Express are really user-friendly, okay? So forget about this. This is how a pipeline look like in like Python, R. So in FCS Express, we really spend a lot of time. Our developers really worked hard to make these type of tools really user-friendly. Indeed, you will not see anything like this, but still you will have all the power that pipeline gives. So the pipeline in FCS Express can be very easily created from the Tools tab by clicking on the transformation. And once you are in the transformation dialog, this is the same dialog that you used in the past if you had the chance to create some K-means, PCA, SPADE, or Disney. Now you have this option, pipeline. So what you see here, when you see here like PCA, SPADE, and Disney, those are standalone algorithms. So those are the algorithm that you can also run in other software. But the pipeline, it's unique, really. So once you have added the pipeline, the main pipeline body is created. Okay, so it's this guy here. In the main pipeline body, you can define the input population by selecting a gate, the input parameters. Okay, this parameter list is retrieved by either a file or a plot. What I usually suggest is to select all the parameters at this stage, because sometimes you do not really know which are the parameters that you want to include. So just include everything at this stage, and at every step you will be able to decide which parameter to keep and which not. But this is just the input of the overall pipeline. Also, if we do not select the parameter here, that parameter will neither be available within the pipeline, but also will not be available as a final output. So, for example, if you do not check the time will not be available within the pipeline, and the time will not be available once you will apply the pipeline to a plot. So this is another good reason to select everything here. Also, the pipeline allows you to select the suffix for the plot title. What does that mean? When you apply the pipeline to a plot, because you want to access the results of that pipeline to that plot, we add a little suffix to that plot. And this is because you may have multiple pipelines. You may apply those pipelines to multiple plots and then compare the results. And if you customize the suffix, it will be easier for you to clearly see which pipeline is applied to which plot. Now, once you have created the main pipeline body, you have access to this drop-down menu that lists all the available steps organized in different categories. You see here we have downsampling step, dimensionality reduction, clustering, visualization, mathematical, and so on. We have like about 50 different steps, and we are going to add more and more in the near future. Now, FlowSOM, it's a pipeline itself. So instead of asking you to create FlowSOM by adding steps one by one, just to make your life easier, we created this predefined algorithm section. Okay, so if you select FlowSOM here, FlowSOM will be automatically created by automatically adding all the different steps that compose FlowSOM. And you can already see here, for example, the first one is a scaling step. Then we have a self-organizing map, of course, minimum spanning tree, the graph layout, consensus clustering. Okay, those are exactly the same step that compose the pipeline, but here in FCS Express are broken down, so you can really tweak each of them independently. And you can change the pipeline and create your own customized FlowSOM algorithm if you want. So let's do this together. I have a layout here, okay, with just a couple of plots. You see here I have a gates, that gates lymphocyte. It's just an example. You can have like 100 plots, 20 pages, multiple gates. And here I have a plot gated on this red gate. Now, let me go to Tools, Transformation. Okay, let's open this drop-down menu and let's click on Pipeline. This is the main pipeline body. This is the same dialog that we saw as a screenshot a few seconds ago. So you can actually change the pipeline name here. If you have multiple pipeline and you want to discriminate them here in this dialog, you can do that. But remember that the actual suffix that would be applied to the pipeline, it's down below. You see pipeline transformed in this case. Now, which is the population that we want to use as an input? Again, this is really up to you. You can run the clustering or the pipeline to all events. You can run it to a specific gate. In this case, just want to show you, I will select a gate from this list. So every gate in the layout is accessible from this list. Then, which is the template file? The template file, it's really just a way to populate this parameter list. Okay, FCS Express otherwise doesn't really know which of the 1 billion FCS file on your computer you want to work with. Now, the template file, just to mention, can be retrieved from a file or from a selected plot. In this case, we just need to select this file, and this is the parameter list. It's the same list that you see here. Okay, nothing to be scared. Now, you can click those parameter one by one. Again, I usually suggest to do right-click and select all. Those will be the input parameter in my pipeline. Now that we have created this main pipeline body, I can go here. And you see we have tons of different options, like downsampling, 5 different types of downsampling, dimensionality reduction, PCA, Disney, and UMAP. Clustering, we have consensus clustering, which is part of the FlowSOM. Hierarchical clustering, K-means, self-organizing map, which is part of FlowSOM. Visualization, graph layout, and minimum spanning tree, those usually goes together and are the 2 steps used by either Spade and FlowSOM to create the nice tree. We have mathematical step. We will deal with the scaling, which is 99% of the time, clustering and dimensionality reduction algorithms benefit from a scaling step to not use the raw data. We also have normalization and 0-1 scaling. You find all the details in the manual. If you have any question, just send us an email. Unfortunately, today we cannot really go through all of them. And anyway, I'm going to go straight and I will insert a FlowSOM right away. So FlowSOM will be added as a mini pipeline, let's say, with multiple steps. And as I was saying, the first one is a scaling step. First of all, why we need to scale? As I said, scaling is usually, sorry, yeah, scaling is usually beneficial for most of the algorithm. There are really very few examples. One is, for example, FlowAI, which really expect data to not be scaled, or otherwise anyway linearly scaled, so raw data. But otherwise, other algorithm are not really happy with linearly scaled data or raw data. And why this? Because for the same reason, you usually apply a scale when you open your fluorescences on a histogram or on a 2D plot. So if you look at your raw data in your FCS file, they are saved this way. Okay, so with linear scale, you see that the negative is really condensed and shrinked here, and then you have a very spread positive populations. So it's not really user-friendly. It's not really easy to define which is positive, which is negative. This is why we apply usually a B exponential, but also a log scale or a hyperlog scale. So we prefer to look at the data this way, where you see the positive and the negative are definitely more comparable and more homogeneous in terms of spreading. Okay, while in this case they are pretty different. So technically speaking, why you need to do this for your algorithm? Remember that algorithm like FlowSOM rely on distances. You always calculate some sort of distance. And if you look at the raw data, okay, look at the variance here, okay? The positive population, okay, cover like, I don't know, 30,000 channel, while the negative are just spread between like 100 channel. So they're not really comparable. Okay, if you want to make distances more comparable, you apply a scale. And this is exactly what we are doing with FlowSOM. And this is why we automatically added a scaling step as a first step in FlowSOM. Now, if FlowSOM is it's like a downstream step in a bigger pipeline and you have already scaled the data before, then you don't really need it. You can uncheck it or you can remove it here with the delete button. But in this case, we only have FlowSOM, so we need that step. You can scale all the parameter and then maybe just use few in the FlowSOM, in the sum, so in the next step, or you can just uncheck the one that you don't need. It's really up to you. How this step works. So if you click on automatic as it is by default, it means that the instrument specific setting that FCS Express uses in your user option basically will be used. So in this case, I'm dealing with a FCS file, standard FCS file 3.0 format. The default setting in FCS Express are to scale the fluorescences with B exponential, scale the time and the scatters with linear scale. So if you select all, they will all be scaled, but the time, the fluoro scatter and the size scatter with linear scale. All the fluorescences with B exponential. So if you want to just scale with B exponential the fluorescences, you can do this, or you can or you can select B exponential, okay, as you want. If you also want to scale the forward scatter and size scatter with like log or B exponential, just add a new scaling step. Okay, you can go here and select another scaling step. And in that scaling step, you will select forward scatter, size scatter, and maybe you will select log or B exponential or whatever. In this case, let's keep things simple. We will scale only the few parameters that I'm going to include in the self-organizing map, and basically are all the fluorescences. But again, it's really up to you. Now, second step is self-organizing map. So the beauty of the pipeline, it's the core advantage of the pipeline, as we said before, is that the output of one step can be the input of the next one. And indeed, you see that in the self-organizing map step, I can access the original data, the raw unscaled, but I can also access the scaled data, okay? And these are exactly the step that I want to include in my FlowSOM calculation. Sorry, in my sum calculation. This is just the SOM step. Now, let's have a look at all these options, because now we know what they means, okay? So first of all, you see here we have these option. I didn't mention that. Just keep them checked. Add meta clustering. At this stage, the SOM, it's not really a meta clustering. Okay, we will see what these two option means later on with the consensus. Just keep them checked as they are by default. Otherwise, you will not be able to access the data. So just keep them checked. Then you see we have really a lot of different options here. So let's try to recapitulate the actual different step in the SOM. So I want to start with the SOM 2D grid. You see you have the width and the height. This is the size, okay? 10 by 10 for FlowSOM. But ideally, you can do whatever you want, 5 by 5, 7 by 7, 20 by 20. Then you have the cluster centroid initialization method. Remember, in the initialization step, you need to define where to place the grid at the very beginning. Okay, we said in the first example, the grid was placed more or less in the center of the data set. In the second example, it was quite far of the data set. You have two options, random, so randomly selected value, or random cells. Okay, by default it's random cells. It means that those 100 cluster 10 by 10 are centered using the same coordinates of 100 randomly selected cells. So the idea is that you're not really starting with random values, but at least you are, let's say, pre-shaping your grid. Okay, so you have an advantage. It's like starting with a grid that already learned a little bit of your data set. Then you have the learning rate. Okay, this is the alpha. Remember the learning, so the movement of every node is defined by the distance between the node and the selected events in that cycle, in that iteration. Then the learning rate, and then the neighborhood function. This is the learning rate. As we said before, the user can define the initial and the final value. Okay, this is basically the default, so 0.05, 0.01. You see, the idea is that the learning rate, it's not really a huge number, like what we saw before, like 60%. Okay, this is much smaller. Then you have the other term, which is the neighborhood function. And we can look at this term here, neighborhood spread. These define let's say, the radius of the neighborhood and the actual neighborhood function, which is this one. Remember, this was either Gaussian, okay, was something like this, or boxcar, something like this. The default is boxcar. You also have here the training decay function, which apply to both the learning rate and the neighborhood spread. The decay function was either these or these, so linear or asymptotic. And indeed, you see that you have both options here. The default is linear. Then you might remember that when you need to deal with the neighborhood function, okay, you always need to, you define the neighborhood spread. Okay, if you remember, the neighborhood spread was, this was our matrix, and let's say like this, and the neighborhood spread was sort of hollow, okay, either well-defined or a shade. But then how to compare each node with this halo? You need to define the distance, okay? And if this is the winner node, you need to define the distance between all the other nodes and define whether some are out and some are in. How to define the distance? There is two options here, Euclidean and Chebyshev. We will not go into much detail. Unfortunately, we always have not enough time during this webinar, but the default is Chebyshev. Last but not least, remember, FlowSOM, it's an iteratory process. So we need to run that calculation. We need to pick up random events from the dataset a lot of time. And how many times? This is defined by the training set size. By default, it's set to percentage and it's set to 1,000. It means that if you have 100 events, you will do 1,000 sampling, okay? If you have like 1 million of events, you will do 10 million, okay, of sampling. The idea is that on average, every event in your data set will be sampled 10 times. Okay, of course, someone will be sampled more and someone will be sampled less, but that's the distribution. Okay, so this is the SOM step. Then we have the minimum spanning tree and the graph layout. We will go through them very quickly. So the minimum spanning tree, it's very easy. You have your nodes, okay? Those are the 100 nodes in your grid. They are distributed along your data set in the multidimensional space, okay? We are no longer looking at the edges that connected them in the 2D grid. Now we are only looking at them in the multidimensional space. The idea is that you will link each node to the closest neighbor this way, okay? So here you see, you calculate the distance between each of them, okay? And then you just keep the one that are closer. And this is the results. So this is the idea. So when you look at the FlowSOM tree, okay, what matters is how those nodes are connected, not actually the aesthetic of this representation. And actually the aesthetic is just aesthetics, and it's determined by the graph layout. So this, what you see here on the left, it's identical to this. It's just aesthetic. So A is still connected to B, B is still connected to A, C, and F, and so on. So there are two methods. The radio layout is the one on the left, and the arch or U-shape layout, just go with the one that is nicer for you, okay? Meta clustering. Just let me go back to the here, okay? We saw the scaling, we saw the self-organizing map, minimum spanning tree, graph layout. You don't need to do much here for the minimum spanning tree and the graph layout. They are just using the SOM cluster as an input. And then you have the consensus clustering, which is really an amazing tool. So the consensus clustering try to solve the issue that comes with the overclustering. Okay, the idea is that in your data set you might have, I don't know, 20 population, 20 biological population, but you're not going to look for 20 cluster. Usually want to look for 100 or 200, okay, 300 cluster. Why do you want to over clusters? Because the idea is that you will shape each biological population with a multitude of small clusters. So it's like increasing the resolution in your camera when you take picture with your mobile phone. But the pain is that if you end up with 100 nodes, as you can see in a spade tree, for example, or in a FlowSOM, then it's really time consuming to go through all of them and say, okay, I think that those are similar. Okay, so we'll draw a line and I will consider those to be a population. And then I think that those are similar, and then I draw a line. Okay, it's much better to do this by a clustering algorithm. So this is the aim of the meta clustering, clustering clusters instead of cells. So you are clustering your cluster in a way that you're grouping similar clusters together. And maybe you realize that your manually drawn gate around the yellow population was not correct. And the clustering is telling you that also those two guys should be included because those are more similar to the other yellow than to the green. So the consensus clustering can be done with every type of clustering. It's really super flexible. Okay, and FlowSOM does that with the hierarchical clustering. So this is the dialog. What consensus clustering is doing, it's really cool. So it's sampling a fraction of your data. You have 100 cluster. Remember in our example, let's say that you sample 90% of them, basically 90 clusters. Okay, this is the sampling fraction. This subset of cluster, this subset of object, it's called perturbed dataset. And the consensus clustering is working with this. You need to define a maximum number of cluster, okay? Let's say 8, 20, 50, it doesn't matter. And then the consensus clustering will try every number between 2 and that maximum number. So if you type 8, the consensus clustering will try to cluster the perturbed dataset of nodes, okay, 90 nodes, with in 2 clusters, 3 clusters, 4 clusters, 5 clusters, till to 8, up to 8 clusters. This is just step 1. This sampling and clustering between 2 and n, it's repeated really a lot of time. You see here, number of sampling 100. Okay, so you are doing all of this 100 times, and at the end the consensus clustering is so good that it's giving you the best match. It's giving you the best, which is called consensus, the best results. And the beauty is that in FCS Express the consensus clustering is so powerful, then it automatically defines which is the best, of course mathematically speaking, the best number of clusters. So if you type 8, it will probably be not enough, because usually you have more variability. But if you type like 50, maybe the actual number of clustering is 34. Okay, so the end final results from the consensus clustering will give you not 50 because you typed 50, but will give you 34, because 34 is the mathematically speaking the more appropriate results. So this is something really unique. It's really a powerful tool, the consensus clustering. And you see that the default is set to be hierarchical, as in FlowSOM. So this is exactly what you see here. Okay, consensus clustering, number of cluster, 90% of sampling fraction, how many times you want to repeat that. So it's a kind of long calculation, but FCS Express is super fast. The very last step that it's added to the FlowSOM pipeline, it's quite optional. It's called new parameter removal. You can find it here under miscellaneous. Okay, the idea is that along the pipeline you may create a lot of different parameters that are required within the pipeline but not in the final plots. Okay, so here we are scaling 5 parameters, and as you can see here, the parameter list here it's longer because it includes both the unscaled and the scaled. At the end, you will also have one more, which is the SOM cluster. So if you want, you can make this list shorter by adding at the very end a parameter removal step and say, I don't want to access the scaled, for example. Of course, you want to access the sum. Okay, that's okay. That's the final goal. But maybe you don't want to access the scaled parameter and make the list shorter. Okay, we are ready to go. Once you want to apply the pipeline, simply drag and drop the pipeline on top of the plots, and you will see that the plot will say pipeline transformed. Okay, another way is to select the pipeline and click on apply to selected, or also apply to all plots. If you want to remove a pipeline from a plot, go to the file, overlays. Let me move these apart. And in the overlay option for that plot, you have a drop-down menu that says whether, whether or not you want to access the pipeline results on that plot. And in case you have multiple pipelines, which are the pipeline results that you want to access. Now you see that we have a new parameter, which is the SOM cluster assignment. Those are the 100 cluster. The 2D representation, it's not really what you want. I just want to show you that the pipeline generate this new parameter. Okay, let's keep it to CD8. What you really want to do is, let me close, okay, is to represent the FlowSOM result as an actual tree. Okay, so just go to insert, plate heatmap, draw an heatmap, and here we go. This is your FlowSOM. Okay, you have a multitude of different options in which you can let's say customize this. First of all, every single node it's colored based on a specific statistic. In this case, it's time arithmetic mean, which is really nonsense. Let's say CD8 arithmetic mean at least. So now you can see that some nodes, the upper left, are red, and all the other one are either black or green. How can you show the different color, let's say, meaning? Just go to format and open the legend, for example. You see we have a legend for the color coding of the fill color of each node. This is defined by the y-axis parameter. In this case, CD8, APC, size 7, arithmetic mean. We also have a color code for the meta clustering. Remember, we looked for 8 meta cluster. And this is depicted by an halo around nodes. Okay, so you see here, you have all the different meta clusters. Now, you can change the shade of color that used to fill each nodes. Remember, they are proportional now to the arithmetic mean of the CD8. You can go to the color levels. One that it's really cool is blue, white, red, is this one. It's pretty nice. You can also add multiple overlays to this FlowSOM. So if you have your data here, I can drag and drop and add as a new overlay. I can do it also multiple times. Let's do it 3 times. Now you see every single node is divided in 3 different slices. And from the overlay tabs, you can define which parameter, for example, to use to color each of those lights. So for example, the first one is CDA, arithmetic mean. The second one can be, let's say, CD4. The third one can be CD19. Okay, now you can clearly see that those are different than these and these and those as well. Okay, in this case, again, let's keep things simple. I will keep just one overlay. So another thing that you can do is to change the radius in this type of representation. You see here in the overlay, you can, by default, it's selected as no radius, okay? But you can change this to whatever, let's say side scatter. And what you see now, it's the radius being proportional to the side scatter arithmetic mean. You can also make it proportional to a lot of other statistics. You see percentile, geometric mean, median, percentages, number of events is usually used. So now you can clearly see that, I don't know, this node contains much more event than this. You can also shape a little bit this node, resize in the specific option. You see, you can make the smaller node smaller, the small node smaller, and you can make the big node larger, okay? Or anyway, change them at your convenience to make a nice representation. Now you can create gates, okay? If you go to gating, well gates, you can draw a gate around one or multiple well. Okay, let's do it. For example, I don't know, these here, okay? And let's say, I don't know, blue and emphasize. And now we see some backgating on our color dot plot. Now, what I want to do, and let me delete this gate, is I don't really want to go and create multiple gates for every multiple nodes or every multiple meta clusters. So what I want to do is to access the meta clustering data generated by the consensus clustering. Okay, you see that so far I just have one single clustering results here, which is the SOM. So let me go to my transformation tool. Okay, and here in my pipeline, now I have access to, I want to show you this option in the consensus cluster, and you will see what's the meaning of this option. We already saw that in the SOM, and it was already selected. Add the meta clustering as a new parameter. I want to do that because I want to access to that results not only as a graphical halo around my nodes, but as actual parameters. And now you see that in my FlowSOM, I can access two different two different clustering results. So I don't want to mess up with these three because I think it's quite nice. I will just open a new one, and I will display the second parameter here. So each of those 8 parameter is displayed as a nodes. Now we didn't create a minimum spanning tree on the consensus clustering. So this is why the nodes here are not organized in a tree, but you can do that if you want. That's the powerful of the pipeline. In this case, they are just ordered at 1 to 8. I can go here, create a multiple well gates. This is one of the new feature of this new version. Let's call this like cluster, and let me flag this option. You see here, create an individual gate for each cluster. Let me do that. Okay. And now you can see that in my gate list, I have now 8 new gates, one for each of those meta cluster. I can also show them here if you want. Okay. Like blue, gray, light blue, green, and so on. And those are automatically backgated you see, in my 2D plots. So this is really amazing. And FCS Express is really fast. Okay, if I move, okay, my gates, let's say that I resize my gates, FCS Express redo the calculation immediately. You see, it took a fraction of a second. Okay, let's do it again. I'm going to move my sales gate. Okay, it doesn't matter if you move it a lot or small, it's still an update. Okay, so it's really amazing what our developers team did with this type of algorithm. Then if you just want to show you the multiplots, if you select, let me zoom out. If you select this color dot plot, you can go to multiplot and create an n by n plot. And in this case, I want to make all the comparison between my parameters. So now you can see here, I have an n by n matrix, okay? And I can clearly see which is which, okay? So I can say, for example, let me zoom a little bit, okay? I can say that, for example, here, these blue are definitely CD19 cells. These light blue are CD4. You can see also here, okay? These green are CD4 dim and CD3 negative, okay? I don't know, monocyte or whatever, okay? I will leave that to you. The gray are definitely CD8. You can see them, CD8. Okay, so this is really a powerful tool. And again, we don't have time to go through these, but you can combine FlowSOM with tons of other tools in FCS Express. So in this case, I just have a predefined layout here in which you see I have FlowSOM, I have an heat map here, I have some spreadsheet, I have all my box and whisker displaying the frequency of my unknown 6 population or 7 or 8 or 10 different meta clustering among different experimental group. And again, this is super fast. So if I adjust this gate, FCS Express will automatically recalculate everything. And you see that also my box and whisker recalculate. Okay, I'm sorry, we are running out of time, so I will just go straight to the conclusion. So in FCS Express, let me clean a little bit. FlowSOM, it's really easy to use because it's just part of the whole user interface. It's accessible to the pipeline, which really gives you full flexibility and full power. It's really fast. You saw it with your eyes. FCS Express, it's really fast. All those algorithms rely on an internally coded we are not relying on like plugins or other software. Everything is coded into the same language of FCS Express. It's combinable with any other algorithms in the pipeline, and it's integrated. So you can use, I don't know, heat map. You can use box and whisker, spreadsheet, regression, whatever. So if you have any question, that's the perfect time to ask that. If you have never used FCS Express, feel free to navigate on our website and click on free demo here at the top. You will access to the 30 days trial version, which is fully functional, all the features, and you will be able to run the pipeline. So really, many thanks. I would really like to stay here a lot of the time more, but yeah, I'm super glad to stay connected for a couple of minutes if any of you want to ask any question. But otherwise, let me really thanks all of you for your patience, for attending this course. If you have any specific interest, if you want to give us feedback and new suggestion, just send us an email. Many thanks.